Assalamualaikum warahmatullahi wabarakatuh. Today, me and my groupmates will be presenting our research on exploring beetle diversity in Pulau Ketam, a morphometric and taxonomic approach. So, you must be asking, where is Pulau Ketam? Now, Pulau Ketam is a small island off the coast of Selangor, Malaysia. It is home to diverse ecosystems ranging from mangroves to grasslands. These ecosystems are vital for sustaining unique biodiversity, yet they face increasing threats from urbanization and habitat fragmentation. Despite its ecological significance, the island remains unexplored, particularly concerning insect diversity. This study focuses on one of the most diverse and ecologically important insect groups, Coleoptera, or better known as beetles. So, according to Hunt, beetles are fascinating organisms with over 300,000 species described globally. Beetles are characterized by their hard, shell-like forewings known as elytra, which protect their hind wings and abdomen. This group includes a variety of species ranging from small inconspicuous creatures to large brightly colored ones. Beetles are found in nearly all environments on Earth and play important ecological roles as decomposers, herbivores, and predators.
Now for literature review for our research. According to the research by Zainul Zamzila and Dawati, and other study was carried out to identify the species of animals found in the mangrove forest of Pulau Ketam, it was found that mangrove forest is a forest ecosystem rich in biodiversity, and this ecosystem is very important in the complex network of life and survival of biodiversity. In addition, the mangrove swamp forest is also a habitat for various types of plant and animal species that can live in brackish soil and tidal waters of Pulau Ketam. Based on this information, we chose Pulau Ketam to be a suitable location for finding beetles. Previous research by James Rolfe and Marcus has demonstrated the utility and effectiveness of combining morphometric and taxonomic analysis to assess the ecological roles and evolutionary adaptation of insects in different habitats. According to a research done in South Africa by McGough and others, beetles are also considered reliable bioindicators as their population often reflect the health and stability of ecosystem. However, there is still limited knowledge of the beetle diversity in Pulau Ketam. This study aims to document the diversity and morphological traits of beetles in Pulau Ketam, addressing gaps in regional biodiversity research.
Next is the methodology of our research. First, we arrived in Pulau Ketam, Perlis at 4 p.m. The equipment was prepared and the search for coleoptera specimens started. Then, areas such as bushes, leaves, branches, and under the rock were examined and explored for 2 hours. Specimen was captured using a bark net and was gently handled to be placed in a secure breathable container. Then, the captured specimen was brought into a lab and was utilized by being placed in a freezer for 10 minutes. After that, the specimen was placed under a stereo microscope. The picture of the specimen was taken by using a digital camera and the observation was recorded. Then, using the picture obtained, morphometric analysis was done on a computer by using an image processing software, and the taxonomy was determined based on the specimen morphology. Finally, taxonomic data was recorded and the morphometric data was tabulated.
Now, for the results of our research, all of our specimens are in the class of Insecta. So the first specimen was Isocoris gratidaris from the family of Entertomidae and order of Hemianthera. So Isocoris gratidaris was identified by its smooth, rounded pronotum and distinct punctation on the elytron. So for the second specimen, was Isocoris literatus, also from the family of Entertomidae and order of Hemianthera. So Isocoris literatus was distinguished by its bright green coloration and elongated antennae, which are common in the bugs. The third specimen was Jonocephalum simplex from the family of Tenebronidae and order Coleoptera. So Jonocephalum simplex, with its robust structure and its dark brown cylindrical body, confirmed its placement within the Tenebronidae family. The fourth specimen was Agolinus entricus from the family Scarabaeidae and order Coleoptera. Agolinus entricus, better known as dog beetle, was identified based on its rounded body, strong mandibles, and densely ridges on its elytra, traits characteristics of species in the Scarabaeidae family. The fifth and final specimen was Saheriscynthus from the family of Sydnidae and order Hemiptera. So Saheriscynthus displayed a combination of black, shiny body coloration and expanded in femora, key identifiers for the species within the Sydnidae family.
So, in this study, we aim to assess the diversity of beetles in the region through both morphometric and taxonomic analysis. However, several factors influenced our findings, leading to a reduced number of beetles collected. One of the primary challenges we faced was the similarity between true bugs and beetles, which led to an error in our initial identification. True bugs, which belong to the order Hemiptera, can sometimes appear similar to beetles, Coleoptera, especially in terms of body shape and size. Due to this morphological overlap, it is likely that some true bugs were mistakenly classified as beetles. The confusion between these two groups might have resulted in a lower count of beetles than that was actually present. The overall low number of beetles observed, however, during the research could be caused by abiotic environmental factors. First, there was a monsoon that occurred just days before our research, and that likely disrupted the local ecosystem. Flooding can cause habitat loss and displacement of insect populations, which could have contributed to the scarcity of beetles in the area. Additionally, beetles are known to be sensitive to changes in moisture levels, and a sudden shift in water levels could have influenced their presence and activity patterns. The high water levels may have also made it difficult for beetles to find suitable places to shelter or forage. Furthermore, the condition of Pulau Ketam's environment played a significant role in the low beetle population. The area was filled with trash, which can alter the habitat in ways that negatively impact beetle diversity. Trash accumulation can lead to a decrease in available natural resources, such as plant matter or decaying organic matter, which many beetles rely on for food and shelter. The presence of food stalls also and a busy road also likely contributed to environmental disturbance, increased human activity and pollution, which can further deter the beetles from inhabiting the area. Insects like beetles tend to avoid areas with high level of pollution or disturbance, and the presence of roads and commercial activity could have further diminished the beetle population.
In conclusion, the study documented the diversity of Coleoptera species in Pulau Ketam, identifying 5 distinct beetles through a combination of morphometric and taxonomic approach. This research also shed light on the morphological adaptation that enable beetles to thrive in specific habitats, contributing valuable knowledge to regional biodiversity studies. Future research should focus on expanding the scope of biodiversity assessment in Pulau Ketam, incorporating seasonal variation and a broader range of habitats. Conservation initiatives must address the growing threat of urbanization and habitat degradation to preserve Pulau Ketam's unique ecosystem. By prioritizing ecological studies and sustainable practice, Pulau Ketam can serve as a model for balancing biodiversity conservation for human development.